hello and welcome back. Today we are starting topic 2. Topic 2 is all about sort of like the basics of everything we're going to cover. So today is all about molecules to metabolism. We're going to focus in on metabolism, what that means, how different macromolecules go through metabolism. But as we continue through this topic, we'll be talking about water, carbs, lipids, nucleic acids, photosynthesis, like the basics of everything that we'll cover moving forward through IB biology. So let's dive right in. We're going to start with maybe one of my absolute favorite scientists and their experiments. So Frederick Roller and the falsification of vitalism. I'm sorry I'm laughing. It's just that this particular story it just makes me a little giddy. So he was a German physician and a chemist, and he believed in this concept vitalism. And what that was is the idea that organic molecules could only be produced within organic organisms. Now, we as human beings are organic, right? We are living things. To be considered organic, that means that you have carbon and hydrogen making up yourself, your organism. Inorganic are non-carbon-based entities. They're non-living things like, a chair or a table. Now, what makes organic things interesting is the use of carbon. So if you've ever been into space, you know that when we discover new planets or identify or go to new planets, we're looking for carbon-based compounds. And the reason for that is carbon can attach to so many other atoms, which allows it to kind of build itself into some sort of living thing. It has the ability to do that. So anyways, back to Friedrich Wöhler. So he believed that organic molecules could only be produced with inorganic organisms, but he mixed two inorganic substances, cyanic acid and ammonium, and then created urea, which is an organic substance. So just imagine for a moment, imagine his surprise. He does this whole experiment and he's like, yeah, this is going to prove vitalism because there's no way that these two inorganic compounds could produce an organic molecule. And then he did it. So it was a total surprise to him, and because he believed in it, he actually is responsible for disproving the idea of vitalism. So I just think that's hilarious, that even though he very strongly believed in this concept, he changed his mind because he proved that that's not true. So he is responsible for both believing in it very thoroughly and then falsifying that theory that vitalism is true. So I just think this is fun. So let's move on. Now that we have an idea of what organic and inorganic is, let's move into metabolism and the molecules that go through it. So the main molecules that we're going to focus on are carbohydrates, proteins, lipids, and nucleic acids. And those are called macromolecules. Those are like the four main macromolecules. If you're into sports or into anything like that, you're counting your macros, that's what that means. It means you're counting how many carbs, proteins, lipids, all of that you're getting. So this chart, you should pause and you should either print out or you should copy down. This is going to be your best friend because you should be able to tell me, if I say, what's the monomer of a protein, you should tell me that it's amino acids, and you should be able to give me an example. So all of this is going to be very important in your understanding of this topic, because we might just talk about, okay, glucose is a what? It's a carbohydrate, but specifically it's a monosaccharide, whereas lactose, still a sugar, still a carbohydrate, but it's considered a disaccharide because it has two carbon rings. So just little things like that. The just basic knowledge of this should be somewhere in the back of your mind, especially for tests and quizzes. So as we continue on, IB does require you to know the actual structure of glucose and ribose. So here you go. Pause, draw, label, all that good stuff. You will have to know this. When I am talking about, these are carbohydrates. When I'm talking about this is a monosaccharide. Mono meaning one, saccharide meaning sugar. But the reason we call it one is because there is one carbon ring here. If this was a disaccharide, like lactose, there would be a second carbon ring over here, but because there's not, we know it's a monosaccharide. Same with ribose, it is a monosaccharide. There are only 5 carbons on this. Every point in the kind of ring that it's making is a carbon, so there you go, okay? And you know that because it says C5. But the carbons aren't labeled in this picture, but it's just every point here is a carbon. Same over here, C6. It's there. Okay. Again, IB does require you to know the basic structure of a lipid. This is a triglyceride. Tri meaning there are 3 legs here. So again, basic structure. You should be able to identify, maybe not draw out, but identify a lipid if seen in a picture. And same with the basic amino acid structure. There are 20 amino acids. I am not making you memorize and learn all 20 amino acids. That is a little bit excessive even for me, but you should know the very basic amino acid structure, and that is what you see on the screen in front of you. So again, pause, draw, learn, love it. This will come in handy, I promise you. Now moving on, we're going to talk about metabolism, what that is, and metabolism is actually made up of 2 different kind of like subtopics. So metabolism is the entirety of catabolism and anabolism. So metabolism is all of the enzyme catalyzed reactions in a cell. And so catabolism is large molecules being broken down, and anabolism is small molecules built into larger molecules. So you can see right here, you have the breakdown, and then you have the rebuilding, and it is a circle. It will continue to be a circle, but all of these reactions are considered under the umbrella of metabolism. Now, when we are making and breaking down organic molecules through metabolism, there are 2 types of reactions. There is condensation reaction or a dehydration synthesis, and that is the building of molecules, right? We have these singular monomers, and they're being built together into a larger polymer. And then hydrolysis reaction is the opposite, where you have a polymer and it is being broken down into individualized monomers. So condensation is going from small to big, and hydrolysis is going from big to small. When we're looking at carbohydrates specifically, condensation reaction occurs when molecules are covalently joined together and water is formed as a byproduct. The bond that is formed in carbohydrates specifically is called a glycosidic linkage. So monosaccharides are single monomers, and when they join together, they form disaccharides, right? Mono meaning 1, di meaning 2. Monosaccharides, when they join together, they are now considered disaccharides. When you join enough of these monosaccharides together, they are considered polysaccharides, which means many. So many sugars, when they link up, they're just called a polysaccharide. The hydrolysis reaction is the opposite of the condensation reaction, so a water molecule is required to break a covalent bond between 2 subunits, 2 monomers. That water will be then kept within those monomers, so water is not a byproduct, but condensation has water as a byproduct. So that is carbohydrates. In lipids, you have a very similar type of reaction occurring, a condensation reaction, but this time in lipids it occurs between the 3 hydroxyl groups of glycerol and the carboxyl groups of the 3 fatty acids. I will show you a picture on the next slide. When you combine this, it forms a triglyceride and 3 molecules of water. When you have this, that bond that is formed is called an ester linkage. I know, I know that there's a lot of vocab, there's a lot of things to learn. We are going to practice this, don't you worry, but these are just like the basics of metabolism that I'm asking you to be familiar with. And then, of course, hydrolysis is the opposite of condensation reaction, just like it was in carbohydrates. So in the presence of water, these molecules will break down into their subunits, into their monomers. So this is a condensation or dehydration synthesis. When you are combining these, right, your glycerol and your fatty acids, right here you see you have two hydrogens and an oxygen. Well, that makes H2O, and so you remove the water, and so you end up with a triglyceride, which looks very much like that picture I showed you of a lipid earlier, right? And you have three waters at the end that are removed from this as you make the lipid. And then finally, condensation and hydrolysis in proteins. So again, it's like all the same, right? Condensation occurs between the amino acid groups of one amino acid and the carboxylic acid group of another amino acid. So again, you have all of this happening. It forms a dipeptide, two peptides, right? Plus water is removed. It is held together by a peptide bond. And eventually, if you get enough amino acids being joined together, you're going to form a polypeptide chain. None of this should be like it's all the same, right? Condensation, you're building. So you're going to have water as a byproduct, and you're going to be forming from monomers to polymers. And then in the presence of water, you have a hydrolysis reaction where polypeptides are broken down back into their individual amino acids. So again, you see this dehydration synthesis or condensation reaction. You have two amino acids. As they join together, you have the OH of one and the H of another combining to release a a water molecule, and now you have you're starting to create an amino acid chain. Okay, I know this is a lot. Metabolism is very molecular, very like nitty-gritty. We're going to practice this a ton, but most of this is going to be like, hey, you need to be able to identify this if seen in a picture, or be able to describe the reactions that I just explained to you over the last like 5 or 6 slides. But I will not be asking you to draw out these reactions with all of the like carbons and nitrogens and hydrogens and single bonds and double bonds and all of that. I won't be asking you to draw it, but you should be able to tell me how it all happens. Okay? Great job. I know this was a lot, but I'm really proud of you for sticking through, and I'll see you in class.